Common exam questions on the A-level Biology AQA syllabus: DNA, protein synthesis, genetic diversity, meiosis and selection. So first one, describe the process of transcription in eukaryotes. Hydrogen bonds between DNA bases break. Only one DNA strand acts as a template. 3 RNA nucleotides align by complementary base pairing. In RNA, uracil base pairs with adenine on the DNA. RNA polymerase joins adjacent RNA nucleotides by phosphodiester bonds between the adjacent nucleotides. And in eukaryotes, pre-mRNA is spliced to form mRNA, or introns are removed to form mRNA. Next one, describe the process of translation. mRNA attaches to ribosomes, or mRNA attaches to the rough endoplasmic reticulum. tRNA and anticodons bind to complementary mRNA codons. tRNA brings a specific amino acid. Amino acids joined by peptide bonds. Amino acids join together with the use of ATP. tRNA, released after amino acid is joined to the polypeptide. The ribosome moves along the mRNA to form the polypeptide. What is meant by the term proteome? The proteome is a full range of, or the number of different proteins that a cell is able to produce. So proteome is all the different proteins that a cell is able to produce. List the differences between mRNA and tRNA. mRNA does not have hydrogen bonds or base pairing, whereas tRNA does. mRNA is linear, or a straight chain, whereas tRNA is cloverleaf shaped. mRNA does not have an amino acid binding site, but tRNA does. mRNA has more nucleotides. Different mRNAs have different lengths, whereas tRNAs are all the same length. mRNA has codons, whereas tRNA has anticodons. Describe the differences between prokaryotes and eukaryote DNA. So, comparisons, similarities are the nucleotide structure is identical. The nucleotide is joined by phosphodiester bonds. DNA in mitochondria, or the chloroplasts, have a similar structure to DNA in prokaryotes. Differences are that eukaryotic DNA is longer. Eukaryotic DNA contains introns, prokaryotic DNA does not. Eukaryotic DNA is linear, whereas prokaryotic DNA is circular. Eukaryotic DNA is associated with histones or proteins, whereas prokaryotic DNA is not. What processes occur during meiosis that increase genetic variation? Homologous chromosomes pair up. Independent segregation. Maternal and paternal chromosomes are reshuffled in any combination. Crossing over leads to exchange of parts of sister chromatids, or non-sister chromatids, or parts of alleles between homologous chromosomes, both of which create new combinations of alleles. Describe what happens to chromosomes in meiosis. Chromosomes shorten, thicken, or condense. The chromosomes associate in homologous pairs or form bivalents. Crossing over forms at the chiasma. They join to spindle fibres, or they're moved by spindle fibres, at the equator or middle of the cell. They join via the centromere. Homologous chromosomes move to opposite poles, or chromosomes separate. Pairs of chromatids separate in the second division, and there is also independent assortment. Describe how meiosis causes variation and explain the advantage of variation to the species. So variation is created by crossing over, independent assortment, independent segregation of chromatids in meiosis 2, or independent assortments of homologous chromosomes in meiosis 1. And the reason it's an advantage is because some can be better suited to their environment or better adapted. Some survive to reproduce, pass on their genes or alleles, which allows for changing environment or a different environment. Define the term exon. It's a base sequence coding for a polypeptide. What is a null hypothesis? It means there is no significant difference between the means of the two populations. That's if it was a t-test. If it was a chi-squared test, there would be no significant difference between the observed and expected values. Chi-squared test. This is to test if there is a significant difference between the observed and expected values. Name the protein associated with DNA in a chromosome. It is histones. Mutation is one cause of genetic variation in organisms. Give two other causes of genetic variation. Crossing over, independent segregation or assortment of homologous chromosomes, random fusion of gametes or random fertilisation.